Thank you, Milan, for that super slick introduction. It's such a pleasure to be back here and meet with all of you guys, and in particular Chad. At the moment, Chad and I share something unique, which is, well, first of all, a passion for oligonucleotides. Second of all, we both have a lingering cold that just refuses to go away. So please watch out for possible explosive eardrum-shattering sneezes, that could happen throughout this presentation. So when I joined Chad's group in 2010, I was a proud polymer chemist, wanting to make a difference in the polymer field. In Chad's lab, I was exposed to nucleic acids, and it's such a magical class of materials that once you touch it, you will never let it go. So that's why in the last 10 years at Northeastern's sister institution, I guess, I've been trying to merge the two worlds, right? Polymer chemistry and nucleic acid chemistry. We had a lot of fun making SNAs that can lighten skin tone. We have made membranes that can capture micropollutants from water. But in the last, I guess, 5 years or so, we sort of settled upon a unique kind of polymer oligonucleotide biohybrid, which are these Bottlebrush oligonucleotide conjugates, which will be the focus of today's presentation. So going back in time, about 4 decades, this is when the idea of using short strands of DNA or RNA to treat disease was first conceptualized. The promise was massive. Any disease with a genetic basis can be potentially treated with a short oligo. And it is possible to use a different sequence to target different disease. So you can potentially think of developing drugs at a much, much lower cost and target a variety of diseases. But ultimately, what has done that was the antibodies. Ironically, the antibodies and oligonucleotides were conceptualized around the same time. And the two modalities really followed very different trajectories. There are more than 130 antibody drugs to date on the market, commanding a massive market covering large disease areas. In fact, for the last 10 years, antibody drugs have dominated the top 10 best-selling drugs every single year. In contrast, the oligonucleotide modality pales in the total sales, in the disease areas covered, and in the number of drugs approved. So this turned out to be, in fact, the comprehensive history of all of oligonucleotide drugs. There's 15 drugs in total. So these stark comparisons really exemplify the kind of differences, the kind of difficulties of turning DNA into therapeutics. So you see, one of the biggest problems is that the oligonucleotide exists in a very awkward size regime. Compared to typical small molecule drugs, oligonucleotide drugs are too large, in fact, to passively across the cell's plasma membrane or the endosomal membrane to enter the cytosol or the nucleus where it can engage with their targets. But compared to typical biologics, such as antibodies, oligonucleotides are too small to remain in the blood circulation after injection. In fact, a big portion of the injected dose wound up getting cleared by the kidney via the glomerular filtration process. And mature technology really only exists to target the oligo to the liver or to the site of pathology by local injection. Developing non-liver targeted, the keyword is non-liver targeted oligonucleotide drugs has been notoriously difficult. Case in point, Exondys 51 is the kind of the marquee drug for a disease called Duchenne muscular dystrophy, which is a rare, fatal muscle wasting disease. The company making this drug, Sarepta, they reported that 70% of the injected dose is cleared from the body within the first 24 hours. What they didn't report is what happens to the remaining 30%. Okay, that data is not available, but we can estimate using the small animal biodistribution data for the same drug modality, which is the PMO modality. And we estimate less than a fraction of a percent of that drug actually goes into the site of pathology, which is the muscle. So the blue circle you're seeing on the screen is actually a pie chart. You can probably make out the thin line here. Let me expand that for you. That represents the amount of drug that actually end up in the place where it's supposed to work. So the very, very low muscle uptake of the material necessitates high levels of dosing, several grams per week for the little boys, 6, 7 years old boys. They need a large amount of this drug dosed every week. So this creates a massive cost of goods issue, limits the therapeutic potential, and makes it very inconvenient for the patient who has to go to the clinic every week just to get the treatment. Sometimes they need to travel 2 hours to the clinic, 2 hours getting the infusion, 2 hours back. That's a whole day wasted just to get a treatment that barely works. The drug enhances dystrophin production from 0% to 0.93% of wild type, so over 180 weeks of treatment. So there's clearly a room for improvement using these drugs. Well, the beauty of the PMO chemistry that Sarepta was using is that these molecules are charge neutral, so they don't stick with proteins, and therefore it is relatively safe to dose the patient in high quantities of this drug very frequently. So there's just enough in the muscle to produce some effect. The same cannot be said for other modifications like phosphorothioates. These molecules tend to stick with proteins and cause a lot of problems. So why is protein binding, non-specific protein binding, a bad thing for these oligos? It turns out these interactions can lead to a variety of side effects that ranges from mild flu-like symptoms to more serious side effects such as thrombocytopenia, kidney inflammation, risk of bleeding, liver toxicity. All of these can be quite serious. So we believe a successful ASO drug has to exhibit improved binding selectivity, meaning they no longer stick with proteins, but they still remain hybridizable to their target. So conceptually, we can do this by encapsulating the DNA into a nano-sized test tube, which has a small opening to allow the DNA to continue to hybridize with the target, but the test tube will block interactions between the DNA with the protein. So how is the industry currently solving this problem? There's a considerable focus on what's called the chemical modification. Basically, instead of using natural phosphodiester DNA, the DNA internucleotide linkage, the sugar, entire backbone or nucleobases can be modified to increase both the cell level potency and pharmacology. But there is a limit to this approach. Although the idea is to find a chemistry that's ideal in every way, but it doesn't seem that's going to happen. On the other side, in parallel, a variety of carriers or vectors have been developed to facilitate delivery. But other than lipid nanoparticles, so far most of these approaches have not gained clinical relevance. And this is not for a lack of effort. More often than not, the culprit is the immune system. So the combined challenges of side effects, toxicity, immune system activation, and delivery really makes oligo only suitable for a very, very narrow disease segment. So here's where we differ from the conventional approach. Our approach is what we call the Brushield conjugate. It's kind of not so imaginative name. Brush is the Bottlebrush component polymer. It shields the DNA, and my wife really didn't like that term. She thinks Brushield sounds like something that you would find in a toilet or something. Well, I said I already filed a trademark for that, so too late. So over here, the blue part is a polymer called polyethylene glycol. It is a proven safe material for pharmaceuticals. And the yellow-pink part represents the DNA. So why is the Brushield conjugate functionally analogous to DNA in a test tube? Well, the dense arrangement of the polymer amounts to an entropic barrier. For the protein to access the oligonucleotide, it would have to reduce the conformational space of the polymer, which makes it less favorable. On the other hand, for DNA to hybridize to the target within the brush polymer, in fact, the DNA hybridization is a volume-reducing reaction. They are made even more favorable in the presence of macromolecules. So we think these kind of constructs will show improved selectivity. So the Brushield conjugates are molecular nanoparticles. In this AFM image, to the left, you can see that each dot represents one of these conjugates. These are highly homogeneous in size, about 20 nanometers in size. They're 300 kilodaltons in terms of molecular weight. If you pass them through a aqueous size exclusion chromatography column, they appear as a very narrow peak, very symmetrical, with a PDI generally less than 1.1. And the particles are near charge neutral, so they don't stick with a lot of proteins, and this is irrespective of the types of oligo that you attach to. And because the particles are not self-assembled, these are molecular species, we have observed no lytic activities. They don't disrupt the cellular membranes whatsoever. So in contrast, lipid-based particles exhibit some level of soap-like activity, and they could lyse up membranes, and causing potential side effects. So here is one simple assay where you can use to demonstrate the selectivity. So what we did is we used a fluorophore labeled oligonucleotide, and we conjugate that to the Bottlebrush polymer, and we hybridize to a quencher labeled complementary sequence. When that hybridization happens, you will see quenching of the fluorescence, and therefore a drop in the fluorescence. But within this cluster of lines, within that cluster of lines, you cannot tell which one is the free DNA, which one is the Brushield conjugate. So they both hybridize extremely rapidly. But if you try to access that double-stranded DNA using a protein, in the second part of the cartoon, you can see the fluorescence restored, but they restore at very different rates. So for the Brushield, the restoration is much, much slower compared to free DNA. So this demonstrates there is a level of selectivity that was imparted to that oligo by the Brushield polymer. Now, one of the main benefits of the Brushield technology is that it provides the conjugate oligo with substantially improved plasma PK. Because the conjugate now evades renal clearance, is sufficiently large, about 300 kilodaltons, and it slows down phagocytic clearance because the PEG is a non-fouling polymer. So the material has much more time to access tissues and cells throughout the body. Whereas free oligonucleotides, either natural or chemically modified, they're cleared very rapidly from the bloodstream, whereas the Brushield conjugate can be retained in the blood for several days. If you measure the area under the curve, the Brushield is much more available, about 2 orders of magnitude more available than the free DNA. So with little renal clearance, the Brushield can now be deposited into a range of non-liver organs. So again, the free oligonucleotides suffers from rapid renal clearance without much accumulation outside the liver and the kidney. The Brushield, on the other hand, shows both increased uptake in non-liver organs as well improved selectivity. So if you're talking about muscle, right, for the free oligonucleotide, the kidney to muscle concentration is 3 logs difference. If you want to raise the concentration in the muscle, the kidney concentration is pretty much through the roof. Whereas if you switch to the Brushield, the kidney is still higher than the muscle, but the difference is no longer 3 logs, it's down to 1 log. So the selectivity is also improved compared to the dose-limiting organ. So the Brushield also improves the retention. So we can see that the Brushield material stays in the muscle for 2 weeks without significant changes in concentration, whereas the free oligonucleotide goes from very low to vanishingly low. Right, so the Brushield helps the oligonucleotide stay where they need to be for much longer. So with this kind of improvement, we can change the biodistribution of Exondys 51 from barely anything inside the muscle to about 15% of the entire injected dose. So that's about a 500 times increase in the muscle concentration. Now, the same trend can be seen by imaging the animals continuously for several months after a single injection. Again, the free DNA is very rapidly cleared from the body, generally within hours, whereas the Brushield materials can remain in the animal for 2 to 3 months. The duration is affected by the oligochemistry. Unmodified DNA can only stay in the animal for about a week, although that's plenty for a therapeutic effect to be accomplished. But if you switch to a nuclease-resistant form of the oligonucleotide, the materials can remain in the animal for as long as the polymer does. In addition, the Brushield materials has a very different biodistribution profile compared with the free DNA. Again, the free DNA rapidly enters the kidney where it's urinated, basically. But the Brushield conjugates exhibits a more even biodistribution. Most of the organs have some level of the Brushield material, including hard-to-deliver sites, such as the muscle, the heart, and the skin. Safety is on everyone's mind, and certainly ours. The Brushield is based upon primarily PEG, which is generally recognized as safe for pharmaceuticals by the FDA. There is at least a dozen drugs on the market that's based upon PEG, protein or peptide conjugates. The material is completely non-cationic, and it's non-surfactant-like. So we haven't observed any form of toxicity of the Brushield in mice after repeated dosage. There's no change in animal body weight, animal behavior, no change in clinical pathology, histological changes to the major organs. There's no cytokine fluctuations, complement C3 changes. What is really interesting in these materials is that they don't produce anti-polymer antibodies, which is really, really unique. So we basically gave the animals 12 doses, either subcutaneously or IV. We dose the animal with the Brushield conjugate or a linear PEG oligoconjugate or a linear PEG peptide conjugate and see which one can generate the antibodies. It turns out anything that the Brushield is associated is antibody-free, either sub-Q or IV, but the same PEG composition in the linear form does generate strong antibodies. So we have some hypothesis of this, right, which is basically that the B cell receptor needs to see the PEG and a co-stimulatory signal, usually that is a peptide or the ASO itself, to generate the antibodies. But because the Brushield is so dense, it would block the B cell receptor from seeing that oligo, and therefore you do not see the anti-PEG antibodies. And this is important technologically because you will want to have a material that can be dosed repeatedly, especially in the context of treating a disease like DMD, where it's essentially a lifelong disease management rather than a short-term treatment. Well, did we generate immunity against other epitopes of the conjugate? Right, so to test that, we measured whether the plasma PK can be affected by repeated dosing. If antibody is generated in large quantities, we would see subsequent injections showing a faster clearance. So we gave the animal 4 doses that are designed to generate immunoglobulins, and then we measured the plasma PK. It turns out the plasma PK is essentially the same, remains unchanged. So this indicates the Brushield is likely able to evade adaptive immunity and does not exhibit what we call the accelerated clearance effect, or the ABC effect. So in the next few slides, I'll be demonstrating the Brushield in a variety of disease settings. And first up is KRAS for non-small cell lung cancer. So as you know, KRAS is the most frequently mutated proto-oncogene in all of human cancer. About 27% of all cancer have this mutation. The protein lacks a deep binding pocket, and it's very difficult to develop small molecule inhibitors for KRAS, and it remained undruggable for 3 decades. This is until fairly recently. A couple of years ago, a small molecule inhibitor for the G12C allele was approved. And the reason that it was able to be approved is that the C refers to a cysteine, which offers the ability to covalently inhibit KRAS. Other alleles still remain out of reach, and this makes it a strong candidate for gene regulation. We prevent the oncogenic protein from being produced in the first place. So we chose a pan-KRAS approach. So instead of targeting a specific allele, we target the wild-type KRAS. This allows us to access, obviously, a much more patient population, access patients whose cancer is driven by wild-type KRAS, and also potentially overcome allele-specific inhibitor-induced drug resistance. So to test the feasibility of the Brushield system, we synthesized a small library of conjugates with different oligochemistries, so a variety of oligochemistries, different linkers, cleavable or non-cleavable, or different polymer architectures. Some of the polymers are linear, some of the polymers are Bottlebrush polymers. So these variations allow us to test the significance of all of these components. Long story short, only the brush polymer architecture is important. All the other features can be tolerated. We can modify them without seeing significant changes in properties. So even the phosphodiester, the regular PO chemistry, works. That's what we're kind of focused upon in the following studies. So we first screened the phosphodiester conjugated Brushield using a panel of KRAS mutant cell lines, including the G12C cells, shown to the top left, non-G12C cells, mouse G12D cells, as well as wild-type control cells. We can see that the Brushield, which is represented by the black curve, shows broad activity against all KRAS alleles, not just the G12C, but also non-G12C mutants as well. In contrast, the commercial drug sotorasib, that was the covalent inhibitor for G12C, shows specific activity towards the G12C cells. And it was better than our Bottlebrush polymer by about 1 log. For control cells, both of these conjugates, both our conjugates and the sotorasib, exhibited very little activity. We think that 1 log difference can be overcome potentially by optimizing our Brushield, the chemistry, the cleavable linker. So I think this still has a lot of potential. We next established xenograft mouse models and allograft models with selected cells. Again, what we found is that the Brushield shows broad activity, a broad tumor suppressive effect, irrespective of the KRAS mutation or the ASO chemistry. Again, even natural unmodified phosphodiester ASO works. But using chemically modified ASO, such as PS or PS-LNA, we can further reduce the ASO dosage by 75% and the dosing frequency about by half to achieve the same anti-tumor effect. So this is the immunohistostaining data that we obtained in the 358 cell line xenograft models. This is the KRAS is basically represented by these brown deposits in the image. We can see that the treatment, which is the third and the fourth image, shows significantly reduced KRAS expression. Initially, we thought this is almost too good to be true. How can you have such a potent downregulation of KRAS across the entire tumor, as opposed to only on the surface? So we had the tumor re-stained by third parties, company, and also the MIT core facility, and the result came back the same. So I think this is really exciting. And these results are really particularly significant when you compare them to the state of the art. So AZD4785, that is a clinically tested ASO from AstraZeneca. In the same animal model, our Brushield conjugate, which is represented by the red and the green lines, shows stronger tumor suppressive activities than the AZD compound. But what's really interesting is that the AZD compound is dosed at 100 times our concentration. So we're using only 1% of their concentration. And the dosing frequency was reduced from daily to weekly. So that's why we say the Brushield can massively increase the ASO bioactivity. They reduce the dosage requirement compared to the state of the art and relax the need for specific chemical modifications. Again, even the non-chemically modified natural phosphodiester, which is the least prone to side effects, works in these conjugates. So while those data are interesting, their potential for further development beyond proof of concept may be in question because the small molecule scene is very, very active. But we also try to dose these materials directly into the lung and see if we can further reduce the dosage, since the primary cancer that we're targeting is non-small cell lung cancer, and so we can try direct dosage into the lung. A main barrier to effective pulmonary delivery is the natural airway mucus, which is anionic material. Right? Most of the nucleic acid vectors are cationic, so they kind of get stuck in the mucus, and this allows them to be cleared by the mucociliary movement. And because our construct is non-charged and non-hydrophobic, we thought it might be a good idea to test direct pulmonary delivery. So interestingly, a single dose of the Brushield delivered to the lung was observed to persist in the lung for several weeks, whereas a free oligonucleotide rapidly crosses the mucus. And you can basically find them in the kidney within 30 minutes, although it's delivered to the lung. This means there is a fairly rapid efflux from the lung. But the Brushield conjugate does not do that. They stay in the lung tissue for much longer. We use a orthotopic model where we dose the animals with a luciferase labeled 358 cells into the tail vein. These cells follow the blood flow into the lung, where they eventually form these nodules in the lung, right? So we can see that the Brushield treatment, which is the second image over here, the Brushield conjugates reduce the number of nodules significantly, and also reduce the size of those nodules. And the pulmonary delivery works even better compared with the IV delivery. So here is the black line is the IV delivery, and the red line is pulmonary delivery. It is more potent, even though the pulmonary delivery received only 10% of the IV delivery. Again, for KRAS and other oncogenes that requires a loss of function treatment, it is generally considered difficult because it is relatively difficult to suppress a oncogene throughout the body, at 80 to 90% efficiency. That's why we're also looking at sort of a gain of function treatments, right? One of these is the cGAS STING pathway. So for those who are not familiar, cGAS is a sensor protein for double-stranded DNA in the cytosol. There's no good reason for double-stranded DNA to show up in the cytosol unless it's a bacterial or viral infection. When the cGAS recognizes the double-stranded DNA, it makes a molecule called cGAMP, which activates the STING molecule, and then that leads to the activation of the innate immune system. Most of the development of the cGAS STING pathway focuses on directly activating the STING, right? The STING agonist. The first generation cyclic dinucleotide, which is a class of molecule that cGAMP belongs to, has already been tested clinically. The application is primarily intratumoral injection, directly injecting these cGAS inhibitors, STING agonists into the tumor. But inside the tumor, the half-life is fairly short. And according to some of the doctors we talked to, sometimes the tumor is so necrotic that you're not even sure that the injection worked or not. So they expressed a strong desire for systemically delivered STING agonist. On the other hand, the cGAP pathway is less explored, and in part the reason is double-stranded DNA is really difficult to be delivered inside to the cytosol. And that turned out to be a strength of our Brushield technology, so we thought we should give it a try. The first question is, how long does the double-stranded DNA needs to be? And this is an important question because the Brushield really does not work very well with long oligonucleotides. The longest that we have tested should be around 30 nucleotide in length. And this is because longer than 30, the oligonucleotide extends beyond the protective shell of the polymer, making the protection less efficient. So we tested a series of length from 20 to 60 base pairs, and you can see that there is a sudden increase in activity, TBK1 phosphorylation, when the oligonucleotide is longer than 60 base pairs. And being double-stranded is important. Single-stranded oligonucleotides does not seem to work. This is reflected in both the TBK1 phosphorylation as well as cytokine expression assays. Is the activation really due to STING? So we created 2 STING knockout cell lines using CRISPR and tested the free polymer, unconjugated DNA, and the Brushield using the 3 cells that we produced. So that's shown here. Essentially, the only condition that can generate TBK1 phosphorylation is using the Brushield conjugate with the double-stranded DNA in the wild-type cells. Any deviation from that shows no activation. This suggests that STING is involved, but is STING exclusive in this case, right? Do you also activate other pathways? If you look at the cytokines, it does seem to be a bit of a non-STING activation as well. But we suspect that this could be from toll-like receptors. But the majority of the activation do come from STING-associated activity. And we did the same studies using BMDCs, and the results were exactly the same. I mentioned that the Brushield only works with small oligos, and you need an oligo 60 nucleotides in length for it to be effective in activating STING. How do you solve this problem? Unfortunately, the 60-mer, when you attach that to the Bottlebrush polymer, shows about the same pharmacokinetics in the blood. That means very rapid clearance from the bloodstream, which is bad news. But we do have a few tricks up our sleeves. The first thing is we can sulfurize the chain ends. That's the blue part in the DNA. Because most of the degradation of double-stranded DNA in the plasma is due to 3' exonuclease. If you sulfurize the chain ends, that kind of blocks the exonuclease activity. So with 3 phosphorothioates, we kind of enhance the blood retention by a little bit. Moving that number to 6, we see a bigger increase in terms of the blood retention. The other trick we have is instead of attaching the oligo at the terminus to the polymer backbone, we can attach oligo using a base in the middle of the strand. So this way, we bring the oligonucleotide much closer to the backbone of the polymer, and this results in a massive increase in plasma PK, which is on par to typical Brushield conjugates that we have observed for other oligos. Does the modifications change the ability of the oligo to engage with C-gas? So we tested the binding affinity of C-gas, purified C-gas with the double-stranded DNA using MST assay, and the binding affinity for the free double-stranded DNA and the Brushield conjugate are exactly the same, almost exactly the same. Actually, the Brushield seem to have slightly higher binding affinity. Does the modifications still allow the C-gas to work in terms of making C-GAMP? So we tested the conjugates with the sulfurized chain ends, the conjugates with the natural PO DNA. All of these were able to activate the TPK1, except for when you completely sulfurize the sequence. When the sequence is completely sulfurized, they no longer engage with the C-gas. But the C-gas tolerates very well the conjugation site, the PS chain ends, no problem. So the long blood circulation times allows the Brushield to enter tumor cells much better and prolongs the retention inside the tumor. We have a half-life about 2 days inside the tumor, and this is plenty to activate the immune system. And this is a B16 allograft model. I think this is one of those harder to treat models. And in the tumor growth profile, this may not be the best tumor response that you have seen for immunotherapy treatments, but we did achieve comparable activity as MSA2. MSA2 is a second-generation non-cyclic dinucleotide agonist. MSA2 was pretty toxic. So basically, in the 2 weeks treatment, only 2 doses of that, the animal body weight was reduced by 15%. In our case, the Brushield conjugate did not change animal body weight at all, and we achieved comparable tumor suppression to MSA2, which again was not so long ago reported, I think, in Science. So this is a major breakthrough. So that's why I'm quite hopeful about this system. We have additional animal models coming online very soon, and hopefully we'll have more data to share in the coming month, maybe in the form of publication. So in the next few slides, I want to very quickly talk about a couple of non-cancer examples. The first one of them is psoriasis. So this picture put up here is probably one of the least disturbing images that you can find about psoriasis. Obviously, this can be a much more significant and severe and debilitating disease. So we selected psoriasis because the Brushield conjugate accumulate in the skin significantly after IV dosage and really distributes across all skin layers. They also enter skin cells, so we tested keratinocytes, macrophages, skin-resident DCs, fibroblasts, adipocytes. The Brushield enter all of these cell lines. And using the EGFP mouse model, which basically produces EGFP protein when you deliver a oligonucleotide successfully to that cell, we can see pretty decent target engagement in the skin that's reflected by this weak band here. Recent studies suggest that the psoriasis strongly depends on the IL-17, IL-23 axis. So we picked a strand that was reported in the literature targeting IL-17 receptor A or IL-17RA. So long story short, switching the ASO to this reported ASO, we saw a pretty potent IL-17RA knockdown in imiquim model or IMQ-induced psoriasis mouse model. The treatment reduced the severeness of the disease. You can see that the cells, the skin of the animal was much less red and much less wrinkled after the treatment. Histologically, that's the image to the right, we can see that the Brushield-treated skin is much thinner in the epidermis compared to the diseased skin, much closer to the normal skin. If you look more carefully, this is Ki-67 stained tissue slices. You can see the number of rapidly growing cells. There's a large number of those in the disease model, much fewer of those cells in the treated group. Once again, what's remarkable about this study is that the dosage we're using is a mere 1% of how people typically dose these materials. We're using 2.5 milligrams per kilo of dosing. And these materials are typically dosed on the order of hundreds of milligrams per kilo to achieve a systemic effect throughout the body. The last example I want to talk to you about is Duchenne muscular dystrophy. This is a rare disease, but quite prevalent among rare diseases. About 1 in 5,000 boys. So the patient gradually lose their muscle over their lifetime, which is shortened to about 16 to 20 years old. With the steroid treatment, they can now last into their 20s, maybe 30s, but the steroids make them grow a little chubby, so it's not an ideal treatment. So the pathogenesis of this disease is that the patients have a mutated dystrophin gene that doesn't allow you to produce the full-length dystrophin. The dystrophin is a protein that links the extracellular matrix to the cytoskeleton. So without that linker, the muscle fibers can literally tear themselves apart. So you see progressive damage of the muscle. The patient's arms and legs are about the same thickness as a healthy adult, but if you do a histology and look at the muscles, the tissues, most of those are not muscle fibers. These are fat and fibrotic tissue. So this is the kind of histology of DMD patients. So fortunately, there is a path to treatment, which is to skip some of the exons and restore the reading frame of dystrophin. So this way, although you will not produce a full-length dystrophin, you produce a truncated dystrophin. That truncated dystrophin is better than no dystrophin, so the patient exhibits a much less severe phenotype. So one of the biggest problem or question, at least for us, is that your construct, the Brushield, is 300 kilodaltons. Do they actually enter the nucleus? Because exon skipping requires the oligo to enter the cell nucleus. And does this big construct prevent that? So we tested these materials in the C2, C12 myotubes, and we do see a pretty decent exon skipping in that case. So these materials, despite the large size, they do go into the cell nucleus. And we also confirmed that the band in the bottom does belong to the exon skipped product by Sanger sequencing. The efficacy may not be as high as the PMO or PPMO, the peptide conjugates that are very hopeful, which are in the clinical trial. But our advantage is concentration. We have, again, 500 times higher concentration in the muscle compared to the free oligonucleotide approach. This is a 14-week study using the MDX4CV animals. So we gave the animals a single 15 milligram per kilo dose. We monitored the exon skipping product using PCR for 14 weeks. The Brushield across all time points, which are represented by these solid bars, they perform better than the B-peptide conjugates, which are these empty bars, which is good news. But we're not seeing 100 times better than the B-peptide conjugate. We're seeing approximately 2 times better. So we have some hypotheses as to why that's the case, and we're trying to make it even better. So stay tuned for our upcoming results. But one of the main advantages of the Brushield is that the pharmacology is dictated by the polymer. So we can conjugate more than one ASO to the same polymer and have them delivered to the same cell. Why is that important? Because, at least for DMD, a lot of the times the patient have a mutation, a point mutation in one of the lesion-causing exons. When that happens, you need to skip the lesion-causing exon as well as a neighboring exon. So you need to skip 2 exons for the reading frame to be completely restored. And it has been very, very difficult to do that because if you dose the 2 ASOs separately, the chance that 2 ASOs end up in the same cell is vanishingly low. But if you conjugate them to the same brush, you can ensure that they're delivered to the same cell. Does this work? So we designed an exon 52, 53 dual skipping construct, and we tested in the C2, C12 cells. We saw a pretty potent band for dual skipping. We also replicated this in vivo using the MDX4CV animals, but this is very preliminary data, only one animal, one time point. But we're expanding this study as we speak, basically in the lab. So hopefully we'll have more to report in the near future. And the 4 diseases I just talked about represents, about a dozen diseases that we have been looking at. So this has been a lot of fun for myself and for my lab. In most of these disease indications, we at least have some level of animal studies that shows potency. So in a lot of these animal models, we see 10 to 100 times increase in potency compared to the state of the art. So we really hope that the Brushield can make a difference in at least some of these situations. The Brushield is not a single material. Any part of the structure can be tuned. So we tested a range of chemistries with regard to the side chain, so that means the PEG, to the linker. Sometimes it's cleavable, sometimes non-cleavable. We tested the different backbones as well. It turns out this part in the middle, where the PEG and oligo are conjugated to, strongly dictates the biological properties of these materials, such as plasma PK, tissue retention, and all of these depends on the backbone chemical composition. So, all right. Each of the Brushield molecule contains a backbone which side chains and oligonucleotides are attached to. We discovered the backbone, again, strongly dictates the biological properties. Given that the backbone can be precisely designed, so that means at each of the position, we can introduce different modifiers. These modifiers could include amino acids, a lipid tail, a cholesterol, etc. Because we can precisely arrange these oligos, these modifiers within the backbone, there is a possibility to probe a large domain of property space. So we think there is what I call the backboneomics approach to these materials. Basically, by tuning these backbones, you can carry out disease-specific optimizations. We haven't completely done that, so again, this is very fresh, so hopefully we'll have more to report. But we did synthesize a section of this library. So all of these, each of the line here represents a different backbone, and these colored boxes represents modifications. And we have synthesized this library. We tested them in terms of cell uptake. In which is the preliminary test, we found that there is a 300-fold increase in cell uptake just by placing a few modifiers that seems very trivial into this backbone in strategic locations. If you do that, the uptake can be massive, much, much higher than, for example, lipofectamine-assisted delivery. Although this construct is entirely anionic, but the fact that they have these modifiers, you can promote the cell uptake significantly. They also changed the biodistribution. So here is a biodistribution profile for a portion of the library that we have synthesized. You see different levels of uptake in a variety of different organs. So we think by using a combinatorial approach, we can eventually accomplish sufficient tissue tropism for these materials. Let's say if you want to target the spleen, we have a spleen targeting construct that takes the materials there at high efficiency. Right now, we're pretty excited about this particular construct here, which is Q36. It goes into muscle about 2% injected dose. The muscle is 40% of the mass, right? So we have a large amount of the injected dose in the muscle. This is for DMD and another disease called DM1. Also, the Q36 enters the cells 10 times higher than the Brushield materials that I showed you earlier. So that's why we're quite excited about that particular backbone. So, ultimately what we want to do is, greatly expand the disease areas that oligonucleotide is amenable to. The field has traditionally centered around rare genetic disorders, which has a very small patient population. With the GalNAc technology that was pioneered by Alnylam, delivered to the liver becomes a mature practice, and this greatly increased the patient population. Right? Imagine the potential if you can accomplish exohepatic delivery to a lot of different organs. Even if one patient benefits from this kind of technology we're trying to develop, I say I'm happy with how my career has turned out. So. I hope you share my enthusiasm for the future of this amazing drug modality, and I thank you for your attention.